now we'll do a brief overview on the different dimensionality reduction techniques that are commonly used in single cell. There are a plenty of different methods that we can be introduced to you. Some of them I'll talk about right now, but there are still other methods that I'll talk during the trajectory analysis, for example, and some of them a little bit later for the data integration. So basically what we have done so far is to gather the reads, do some QC, get the raw counts and the count QC so far. But the workflow for each data set is quite specific, then depends on the research question, the batches, experimental conditions that you have, sequencing method, and so on. So then now we got also the normalization step. And from that, you do principal component analysis. And from that, you will do TSNE and UMAP. And PCA and TSNE and UMAP are dimensionality reduction techniques that I'll talk today. And from PCA, then you also do clustering and then differential expression with the clusters. But there are many ways that you actually can do that. So you can go directly from normalization to differential expression. For example, if you already sorted the cell types that you want to compare, then you don't need to do clustering because you already know which cell types they are. And there are many ways that you can do. You can do the PCA directly to differential expression. Normalization, you can also compute the TSNE and map directly from here without PCA. There are many ways that you can do, but these ones in black are the common way of doing that. Yeah, so you can also do clustering from TSNE and UMAP, or do differential expression or further analysis with the normalized data. But why do we need to do dimensionality reduction? So we've seen a little bit before some mentions about PCA. So the idea is, the main idea is to simplify complexity, so it becomes easier to work with. And there are many purposes for that. So imagine that you have then your number of samples and n amount of genes, and then you want to reduce the dimensions for just 2 dimensions in this case, and that you can use for example, visualization. And in some cases, you want also to transform non-linear relationship between cells to something linear. And that is done usually using a graph that I'll mention a little bit later in this lecture. Another way is to remove redundancies in the data, because you have 20,000 genes. Most of those genes will actually have exactly the same expression trend. So some cells will have the same expression of a transcript because they are the same cell type. So you actually don't need all that information from all those genes to show that those cells are similar or different to each other. And in that way, we can identify what is the most relevant information in your data set and filter noise, and that can be done using PCA, for example. So reduce the computational time, so you have less data to work with for downstream procedures. Facilitate clustering, because many cluster methods will struggle with if you have too many dimensions or if you just cluster on the raw data. So if you find first what is the most relevant information in your data and then you cluster on that, then you will likely have more robust results, and your clustering algorithms will also perform much better. The same thing goes to the selection of which genes are actually important for your dimensionality reduction, such as selection of the highly variable genes, as also mentioned. And the latest purpose would be data visualization as your common use to that. So either using TSNE or UMAP, that I'll also explain today. And there are many other reasons, but these are the most important for single cell. So dimensionality reduction algorithms can be divided into 3 major groups. So matrix factorization based, so it's a linear algebra multiplication of matrices to find a reduced space. Graph-based methods and autoencoder-based methods, which can be considered as a part of the matrix factorization as well. There are many different methods, some of them also being developed specifically for single cell data analysis. So today I'll talk mostly about PCA, which is very, very, very well used. So here, for example, I can see ZIMWave that was already mentioned. So it's also considered a way of dimensionality reduction. TSNE and UMAP are also the most used ones. TSNE is now getting in disuse, but and is being replaced by UMAP, but both are still quite widely used in the literature to this day. Basically, some of those methods are adaptations of the PCA. So for example, sparse and an NMF. So it's negative matrix factorization and is sparse notation because it's single cell. So you account for the dropouts. And some of those methods are similar in that way. So it's an improvement over PCA, for example, or for multidimensional scaling. The same thing with the diffusion maps, large Vs. They are somewhat similar to TSNE and UMAP. So that's why I'm not going to so many details on those right now.